used to work with Kulkarni Endowment. Yeah. Who's also a chemical engineer who worked with the Lightfoot. Sure. As most of us have. Yeah. So shall we begin?
Very good morning to you all. I feel honored and privileged to welcome you all on behalf of CSIR-NCL to Dr. B.D. Kulkarni Endowment lecture today. A little bit about NCL before we start. National Chemical Laboratory, NCL, Pune, was established in 1950, and it is a constituent laboratory of the Council of Scientific and Industrial Research, CSIR. CSIR is a science and knowledge-based research, development and consulting organization. It is internationally known for its excellence in scientific research in chemistry and chemical engineering, as well as for its outstanding track record of industrial research involving partnerships with industry from concept to commercialization. A few mission statements for NCL are that it is to carry out R&D in chemical and related sciences with a view to develop eventually and deliver a product, process, intellectual property, tacit knowledge or service that can create wealth and provide other benefits to NCL stakeholders. So a little bit about Dr. B.D. Kulkarni's Endowment Lecture Series. So the NCL Research Foundation, which promotes excellence in science and technology, has instituted the Dr. B.D. Kulkarni Endowment Lecture Series in 2023 to keep the cherished memory of Dr. B.D. Kulkarni alive. The NCL Research Foundation is supported by a corpus of generous donations made by individuals and companies. It is registered as a non-profit public trust and managed by a distinguished board of trustees drawn from academia, public life and industry. So who was BDK? To us all, friends and colleagues, he was known as BD. But Dr. Bhaskaran Dattatreya Kulkarni, 1949 to 2019, completed his BTech and MTech in chemical engineering from LIT Nagpur, currently known as Lakshminarayan Innovation Technology University Nagpur. He started his research career at NCL as a PhD student and continued there till his last day, with formal employment spanning from a junior scientist C to the DG level distinguished scientist. His work significantly embellished every area that it touched, from mathematical analysis of stochastic reactions, nonlinear dynamics to chemical reaction engineering. Data-driven modeling, noise filtering, chemical and biochemical process design, scale-up and commercialization were parts of his repertoire. BD was recognized with several awards and distinctions, a few being the S.S. Bhatnagar Prize, membership of the Indian National Academy of Sciences, of Engineering, the IAS, and the TWAS, and also the renowned J.C. Bose Fellowship. His scholarship and gentle mentorship made him an invaluable asset to the entire chemical engineering community across India. So it is with great pleasure and privilege that now I welcome Professor Mark Prausnitz here today. Dr. Prausnitz is a Regents Professor, J. Erskine Love Junior Chair in Chemical and Biomolecular Engineering, Director for the Center of Drug Design, Development and Delivery at the Georgia Institute of Technology, Atlanta, Georgia, in the United States. I am delighted to introduce you to the groundbreaking work of Dr. Prausnitz, his dedicated team. Dr. Prausnitz is a pioneer in the field of biophysical drug delivery, revolutionizing the way we administer medicines, vaccines, and even gene therapies. His research focuses on harnessing the power of microneedles, ultrasound, lasers, and other physical forces to precisely control the transport of these vital substances into and within the body. His cutting-edge approaches have the potential to transform healthcare by making treatments less invasive, more effective, and more accessible to patients worldwide. So one of Dr. Prausnitz's most promising innovations is the development of self-administered microneedle patches for vaccines. Beyond vaccines, he's also interested in exploring the use of hollow microneedles for targeted drug delivery in both the skin and the eye. So imagine insulin delivered precisely to the skin for rapid onset in diabetic patients. His research delves deeper into investigating how physical forces like ultrasound and laser-activated bubbles can temporarily open cell membranes and allow for delivery of proteins, DNA, and other molecules into the cell. So Dr. Proxnitz is not only a brilliant researcher, but also a dedicated educator and a mentor. He shares a passion for science and engineering with students through courses he's developed on pharmaceutical sciences and technical communication. Finally, on a personal note, it's fascinating to learn that Dr. Proxnitz's work builds on a rich legacy of scientific excellence. He's a student of the renowned Professor Bob Langer, who just received the Kavli Prize from MIT, and his father, Professor John Proxnitz at Berkeley, was a pioneer in molecular thermodynamics. He's also a successful entrepreneur, co-founding 10 startup companies, including one with Dr. Saad Bamla, who was actually one of our very own. He was an intern at NCL, and he's currently now a professor at Georgia Tech. So please join me in welcoming Dr. Proxnitz to the stage to deliver the Dr. B.D. Kulkarni Endowment Lecture with a very benign title, Development of Microneedle Patch for Self-Administered Long-Acting Contraception, but with very far-reaching implications. Dr. Mark, the stage is yours.
Thank you so much for that very kind introduction. Just want to make sure, is the microphone working okay?
Yeah.
Yes, it is. Yes? Okay. So I'd like to tell you about some work that we've been doing. As mentioned, so I come from Atlanta, Georgia, in the United States. Some people know Atlanta because it's the home of Coca-Cola or the home of CNN, but we like to think of it as the home of Georgia Tech. And so I'll tell you about the work that we've been doing there. As mentioned, we're working in a variety of areas of leveraging technologies generally developed for other purposes and adapting them, repurposing them, coming up with ways to use them for medical applications. And the microneedle is the most central concept that we work with, and it can be manifested in a variety of ways. And what I'll tell you about today is how we're using it for self-administered long-acting contraception, and I'll describe why we think that should be of interest. So Georgia Tech does require that I mention that there are companies that I've helped to start, and so I have a financial interest in some of the technologies that we're discussing. The work was supported by the USAID and the Gates Foundation through another organization called FHI, so let me just give the acknowledgement for that funding source right up front. So here's the motivation. Contraception is, of course, a very important topic, and there are, as a result, a whole variety of ways in which contraception can be accomplished. And if we look at the very top, we have methods that are the most effective methods, and those, of course, sterilization clearly works well, but
it's fine.
But in addition, then, the other ones that work very well are the reversal methods that are long-acting. So maybe an implant that is put into the body or an IUD that's in the body. And the reason why they can work so well is that they don't require the user to do very much, right? They have to go occasionally, but then everything is taken care of. And then as you look further down the list in terms of things that are effective, they require more activity. So to take a daily pill, for example, so that requires that a woman remember to do that and do it reliably, and it doesn't always happen so reliably. And then the least effective ones are the ones that require activity right at the time of intercourse. And so the less invasive into a person's lifestyle the method is, the more likely it's going to be used effectively. So with that thinking in mind, although there are so many methods that exist, what's really missing in this landscape is something that has a combination of two things. One is long-acting, so that's with the great efficacy and convenience of a long-acting method. But the other co-word that goes with that is self-administered. Usually the self-administered methods are things that you have to do on a frequent basis, and so they don't provide that reliability. And so our objective here is to have the self-administered capability along with the long-acting capability, and that doesn't exist today. So some further motivation on this topic is that there are more than 100 million unintended pregnancies around the world every year, and Actually, I'm sorry, this is just the number that's in low and middle income countries. It's, the worldwide number is even more. And 77% of the women in these cases report an unmet need for contraception. So they're not, although there are many contraceptive options, they're not adequate for the needs that the majority of women have. And so the contraceptive limitations are, there's a lack of user adherence, there's a lack of discrete contraception, and for some cultures that's important, especially important. There's a lack of effectiveness, there's a requirement for professional administration, and as I mentioned, there's a lack of non-invasive, long-acting contraceptive methods. So objectives that we have are to develop a long-acting efficacy with a single application, non-invasive, discrete, self-administered, and low cost. And our solution to that involves a microneedle patch. So if you want to get a drug into the body, you might take it orally, and that's going to inherently be something short-acting because the GI tract moves everything through. Or you'll inject it or implant it if you want something to be longer-acting. We think that we can accomplish that goal of putting something into the body, not via the oral route, if we make a needle, but make it very small. Make a micro needle. And here you can see a picture, so with little magnification and then greater magnification of what a microneedle patch looks like. So these are microneedles that are on the micron length scale, so typically hundreds of micron length scale. And so they can carry a drug into the body, but do so in a way that isn't like an injection that requires a doctor or a nurse, but is a patch that you simply apply to the skin. As shown here, and the idea is that we can take this microneedle patch, press it to the skin, and unlike other conventional patches, maybe you know the nicotine patch or other patches that exist, you don't wear it a long time. The patch is just a way of applying to the skin, and as shown in the cartoon, the needles separate from the backing, so that when you pull the patch off just a few minutes later, the needles have remained embedded in the skin and can release the drug, either quickly or, in this case, slowly over time. So to explain the challenges a little bit further, here is some anatomy of the skin. The very upper layer, which is the pink at the very top, is called the stratum corneum. The stratum corneum is just 10 or 20 microns thick on most of the body, and that is the main barrier layer. That's why when you put something on the skin, it doesn't enter into the body. And generally, that's a good thing. We don't want any random thing that lands on the skin to go into the body. But when you want to do drug delivery, it's not such a good thing. So that's the barrier we need to overcome. Below that is the viable epidermis. That's where you can see all of the purple structures. So those are the nuclei of all the cells that are filling the viable epidermis. The viable epidermis does not have blood vessels in it. So in dermatology, that's a good target, but generally if you want to deliver a drug systemically to the whole body, you want to reach the blood vessels so the drug can be taken up, and that's why you need to get then into the dermis, which is the bulk of the skin. Viable epidermis is just maybe 100 microns thick. So if you can get deeper into the skin, you reach the capillaries, and then a drug that is being released can go into those capillaries and go throughout the body. So our goal is then to make some microneedles that are able to cross that stratum corneum barrier, and even better, get all the way across the epidermis to reach the superficial dermis. I mentioned there are no blood vessels in the epidermis, so there's a lot of blood vessels at the very upper dermis to provide nutrients to the cells that are in the epidermis. So if we can reach that superficial dermis, we can reach the blood vessels and deliver the drug systemically. Microneedles do take a whole variety of forms, and depending on what your goal is, you might use a different kind. So there can be solid microneedles that are just piercing structures. So you press the microneedles into the skin, you pull them out, and now there's some micron scale holes in the skin. The holes are microscopically small, you don't feel the needles when they go in, so it's a painless process. But as far as any molecules are concerned that you want to deliver, these holes are huge. Any drug you might think of is nanometer dimensions. The holes that we make are micron dimensions. So they're small to you and me, but they're very big to the drug molecules, and they can now be absorbed more effectively. We might, rather than letting the drug slowly diffuse through some residual holes, we could coat those needles with the drug. And now you can press the needles on, the coating dissolves off very quickly, maybe even as short as a minute, you pull the needles back out again, and now you have a very simple and rapid method to administer the drug and take the patch away. We could make the whole microneedle out of drug and other material that encapsulates it. It could be a water-soluble material, and so we'll often use water-soluble polymers like carboxymethylcellulose or polyvinyl alcohol, and often there'll be some sugar in there like sucrose. So you can press those microneedles in, the whole needle dissolves, so there's no sharps waste when you're done. Or, as you'll see in the application we're primarily interested in today, we can make it out of biodegradable polymer. So it doesn't quickly dissolve, but it can slowly degrade and dissolve and release a drug over a longer period of time. There is finally, rather than the solid needle, there's the hollow needle concept, more like a hypodermic needle. You have a liquid formulation, you inject it in through the hollow needle. Our focus will be in this dissolving microneedle, or really biodegrading, followed by dissolution once the degradation process has occurred, for slow delivery. So here's an example of what a microneedle patch can look like. These are the kind that we will often use. You can see 10 of them here in the foreground, needle and syringe in the background. And if we take a closer look at one of these patches, you can now see this has an array of 100 microneedles, 10 by 10. Each one has in it some green dye to help you visualize it. There's a shiny area around the periphery. That's some adhesive, so when you press it to the skin, it'll stick for the period of time that you need it to. There's a tab that you can grab onto that doesn't have adhesive. You can make it in different ways, but this is one design that we like. We can take a closer look at those microneedles, and you can see a little better what they look like now. So these are the solid microneedles. In this case, we have selectively loaded the needle tip portion with the green dye, representing where the drug would be. The backing does not have it, and that's important because the part that enters the skin is what you want the drug to be in. You don't want it in the backing. That would not be productive because that wouldn't be delivered. And another picture shown here to give you a sense of scale. So some things that came into our thinking in developing this technology are, first of all, patient administration. We would like these patches to be self-administrable, and indeed, you just press it to the skin with your thumb and you peel it off afterwards. So essentially, no training is needed. Anyone can do it. There's no applicator involved. Your thumb is the applicator. There's no reconstitution. So that means sometimes a drug may be in a dry state, say in a vial. You add water and dissolve it, and then give the injection. So that's the reconstitution process. We have a dry formulation, and that can give extra stability and other advantages. But we keep it dry. We deliver it in a dry state. Painless delivery, no fear of needles. Again, facilitating people self-administering, facilitating access and acceptance. The manufacturing method looks like this. So we'll start out by making a mold in the inverse shape of the microneedles. The mold usually is out of PDMS, so silicone polymer. We'll make that off of some kind of a master structure, which in the olden days we etched silicon wafers. These days we can 3D print that master structure, make a PDMS mold off of it. We then do a first cast into the mold, which has the drug in it as well as whatever other materials. In our case, it would be the drug plus polylactic acid, the biodegradable polymer. We then allow that to dry. We then do a second casting process to make the backing. The backing does not have the drug and would be, in our case, it would be out of water-soluble materials, so that when we press the patch to the skin, the backing part can at least partially dissolve, and then the needles are released and remain in the skin. So once that whole thing is dried, then we can remove the patch. So the manufacturing process is, at least conceptually, a simple one. We prepare 2 solutions, we cast 2 films and dry them, and then put into a package. As you might guess, there are some important details to getting this process right. What really is quite critical is controlling the transport processes that are taking place within this microneedle cavity of micron dimensions. You have to get it to fill properly, you have to get it to evaporate properly. Solvent can be pulled into the mold as well, so solvent's leading into the mold as well as out into the air that surrounds it. And so that turns out from the manufacturing perspective to be the really critical piece, is controlling those transport processes taking place within the mold to get the needle to take the shape you want. Transportation and storage is facilitated. Obviously it's small, and so that is beneficial in a variety of ways, and we get some improved thermostability We do work with vaccines, and that becomes quite important for thermostability. Contraceptive drugs are generally stable already, and so we don't have to work too hard to keep them stable. And finally, from a waste disposal standpoint, there are no sharps waste. The microneedles here, this is in the case of the ones that dissolve. As you'll see in this talk, we'll emphasize more ones that separate, but in any case, the patch that's removed has no needles on it. So you throw it in the trash, you don't have to worry about safety issues with sharps waste, and any kind of a reuse is basically impossible because there's nothing left to reuse. So let me go through a few studies that we have done. In most cases, studies that were published, and so this was our first foray into this field of using the microneedle technology for contraception. And you'll see rapidly separable microneedle patch for the sustained release of a contraceptive. So the rapidly separable microneedle patch, the key is we don't want someone to wear the patch for up to, say, 6 months. We want the patch to be worn briefly, the needles separate, and then you throw the patch away. And then sustained release is the other really key word here, that we want that release to last for a long time. You'll see our initial objective was 1 month, and we'll get to 6 months. So the concept works like this. You can see the design of the microneedle shown on the left. We have a microneedle made out of a combination. At this point, we had polylactic coglycolic acid as well as polylactic acid mixed together with the levonorgestrel, which is the contraceptive hormone, and that all creates the microneedle portion. We then put a bubble, an air bubble, between the microneedle and the backing. That facilitates the rapid separation, as you'll see. And then we have a backing layer just to hold it all together. You can press those microneedles into the skin. You then apply a mild shear force. The shear force causes the microneedles to break, so the microneedles are sufficiently strong to be pressed into the skin. But because there's that bubble there, when you apply a shear, the needles snap off immediately, and then you can pull the backing away, leaving those needles embedded in the skin. The way that we make them works like this. So we start out with this silicone mold, and we do a first solution that gets cast on. That solution has in it the drug and the biodegradable polymer in organic solvent. Cast that into the mold and fills the needle cavities. We then do a second cast, which is an aqueous layer, an aqueous cast, and so they don't mix, so that's very nice. We keep a nice clean boundary between the microneedle, which was made of hydrophobic materials, and then the backing, which is hydrophilic, and then let it dry. In fact, the fact that the first cast is hydrophobic and the second one is hydrophilic is actually quite important, because remember, we want an air bubble. So there is poor wetting of the second cast onto that first cast, and that poor wetting, there is some wetting that occurs, so there is a connection that's created between the backing and the needle, but it's an imperfect one, and air bubbles can be incorporated as a result of that. So I'll say a little more about that in just one moment, but here you can see a picture of some of these microneedles, and the magnified view with the arrow shows where the bubble is located. This is a relatively large bubble. We can make them of different sizes, as you can see here. And the way that we control the bubble size is very simple. It's based on gravity. And so if our second cast solution, if we cast just 30 microliters of backing solution, we get a bigger bubble because there's just less mass of the aqueous solution pushing down and forcing the aqueous solution against that hydrophobic surface of the microneedle. Or if you put 90 microliters on, then there's a greater force and the bubble gets smaller. So a very simple method to tune the bubble size, and you can see then the graph showing that relationship. So, okay, so now we've made some of these microneedles and we've added some dye into them. So this is a sulfurotamine, just a fluorescent dye to help us image the biodegradable polymer portion of the microneedle. We can press them to the skin, apply that shear, pull them away, and now you can see the microneedles are gone. Just the backing layer remains, but where the bubble exists, that interface has broken off. And then if we look into the skin, you can see all the microneedles embedded in the skin. This is in pig skin ex vivo. The dye is red. There's no blood involved. Green dye is probably better. This is the microneedles embedded in the skin. So then we went from ex vivo, we moved on to the in vivo model. So now we went to the rat. And you can see here the rat where the patch has been applied to the skin. If we take a closer look at the skin, again, you see all the little red spots in there where all of the dye needles have been placed. And if we do histology on the skin, so take a cross-section through the skin, you can see a couple of those needles embedded just below the skin surface where they've broken off. We can follow that then over time. So we are darken the room and are using fluorescent excitation to see the microneedles in the skin with a fluorescent dye. So day 0, right after the needles have been placed, you can see all 100 of them are embedded in the skin. And then over time, it dims. And we're not seeing that whole needles are going from bright to disappearing, and suggesting a needle fell out, for example. But we're seeing all the needles are there, they're just getting dimmer and dimmer as they biodegrade and as the dye is being released. So it looks like we have successfully implanted those microneedles in the skin. They're staying put, and they are releasing a dye over time. Ultimately, we want to release a drug over time, so here's that result. So now we made these microneedles with the levonorgestrel contraceptive hormone in them. The very same thing with those rats. And you can see, well, the two lower curves are uninteresting. That's either blank microneedles, no drug, or just nothing done to the animal. So those are the background controls. But what's more interesting is the black data points, and that is for the microneedles that have the contraceptive in them. And you can see that we are having a sustained release over time. The dashed line represents the human therapeutic level, and these are rats, of course, but just as a target point for us in this study, where you can see for about a month, we are maintaining the blood levels above that target level, and then for a few weeks after that, it tapers off below. So this suggests that perhaps this would be suitable to be used as a monthly patch. Apply a patch on, then do nothing for a month, and then the next month apply another one, enabling a woman to have the long-acting, at least month-long contraception with a simple self-administered patch. We can back out, using a pharmacokinetic model, we can back out this concentration over time data into the amount of levonorgestrel being absorbed or released out of the patch over time, and you can see characteristic kinetics. This is our first-order release kinetics, which is initially fast and tapers off some. So that then led us to a next variation in the process, and that is, rather than having the bubble as the way to separate the microneedle from the backing, we didn't like the bubble so much, in part because it required a shear force. And so you then have to either instruct a person, press, and then slide your thumb to the side, and they may mess that up, or you could build a little device that could enable the person to by pressing it, would sort of automatically slide your thumb, but we thought maybe another way would be good, and so we came up with using effervescence. Rather than put a bubble in initially, create the bubbles through effervescence, and I'll show you what that, how that manifested itself. So the basic design is the same, but we don't have a bubble, and instead that backing material between the needle and the rest of the patch, that has effervescent material. That is, it has, let's see, got the reaction. So it uses a sodium bicarbonate and citric acid, so classic effervescence chemistry, and when that gets wet, then you get chemical reaction that makes carbon dioxide bubbles, and that then can weaken the interface between the microneedle and the backing, and then you get the separation. So we made some microneedles like this, and you can see some of them here before in air. So the pink part is the biodegradable polymer part with the dye, and the part below it is the water-soluble part that has the sodium bicarbonate in it. As soon as you drop it into saline, then the bubbling begins. I've got a little time course here showing that bubbling occurring, so 0 seconds, and immediately it starts, and within a few seconds, the needles that are buoyant, so they're less dense than the water, you can see them, the bubbling has occurred enough that separation occurs and the needle floats up to the top. There's not quite as much water in skin as in a bath of saline, so the kinetics are a little bit slower in the skin, but we believe basically this is what's happening when you apply the patch to the skin as well. And we can use microneedles like that, and we generate a curve, a pharmacokinetic curve that looks a lot like the one you saw a moment ago. Not surprisingly, the microneedle itself was minimally changed. It's just the backing and the separation mechanism. And I should clarify, when these are pressed to the skin, they're left in place for 1 minute. So we don't have a 1 second separation time like we have with the bubble. We have to wait a little while for the effervescence to kick in, but we think that should be okay. Somebody can leave it on for a minute and then take it off. And if you leave it on longer than a minute, it shouldn't be a problem. Less isn't so big. So we then applied some of these patches to human subjects to see how that worked and what people thought. In this case, the tip portion of the microneedle was not made of biodegradable polymer, did not have drug in it. It just was made out of some water-soluble materials that would be safe. We didn't want to make implants in people just for this study. We used placebo patches, and the main point was to use the microneedle geometry with the effervescent backing and see how that worked. So you can see after applying these to human subjects, and these are all women of reproductive age, you can see the needles before, and then afterwards the needle tip is gone, and that needle base looks kind of messy. And so that's because the effervescence has occurred, and so it's not a nice smooth structure anymore, but it's been distorted by that effervescence process. We can then look at the skin, and this is a little different than the red microneedle pictures I showed a moment ago. In this case, the microneedles have no color. We've applied a dye to the skin called gentian violet, and that stains the sites where needles have penetrated. So again, with humans, we want to be very careful. We didn't want to deliver a dye to the humans. Gentian violet, though, is used actually as to treat skin wounds. So it's actually a drug that is used, but we use it just as a dye. You can then look at the skin, so we applied them to the wrist in this case, immediately after application, an hour and 24 hours. And what we saw here, which is what we usually see, is a very faint erythema or redness where the patch has been applied, and it's punctate. So you look closely and you see individual little dots. So the micro injury of each needle going into the skin caused a highly localized and again very mild erythema. You can see it if you look very closely, but at a distance you wouldn't even notice it, and that then resolves itself usually within a few hours, definitely within 24 hours. No medical or safety significance, just a minor cosmetic significance to having a brief redness. So that led to a third generation of this separation mechanism that I'd like to describe. And so we didn't like so much having the effervescence throughout the whole microneedle, and we wanted just to have a thin film of that effervescence in order to facilitate the manufacturing process. And so here you can see schematically the objective here is again to have this effervescence generated at the base of the microneedle. So a little more information about how we now made these. So this is similar to what I'd shown before. We have the mold, and we put, in this case, it was a PLGA with levonorgestrel solution in to make the needles. And then we make a backing that has polyvinyl pyrrolidone and sucrose in it and then demold it. So if we make these microneedles with this porous polyvinyl pyrrolidone and sucrose backing, I guess I should clarify. This doesn't involve effervescence now. This involves a porous material, and we create that porosity through lyophilization or freeze drying. So when you freeze dry something, you first freeze it, and so you fix the material in place, and then you dry it, you remove the water. And because you have frozen it, you don't get a collapsing of the material down, but rather wherever the water gets removed, you get voids, you get porosity. And that's actually nice if you have a drug in a vial and you want to add water to it and have it dissolve very quickly. You'd like to have lots of voids so the water gets in. That's not our goal here. Our goal is to have those voids so that they're little air pockets, and we have made this mechanically weak. So we don't have a big bubble, and we don't have the effervescence. And a challenge with effervescence is that if any humidity gets into your package, it starts effervescing. And we don't want it to effervesce early. We want it to effervesce once it's in the skin. So this avoids that complication. All right, so you can see now some of these microneedles that have the solid without the lyophilization, and then the porous version of it where we did lyophilize, and you can see the porosity that was generated. So we then compress those microneedles into the skin. So this is the control group, not porous. Press into the skin, stain the skin. You see the blue dots, the needles went in. We then look at the microneedles afterwards, and you can see that they didn't detach. When we look at the skin, we see very little red. Red is the fluorescent dye. So they punctured, but then they came back out again. And there's the use patch where the needles are still there. And so if you look at the same thing now with the porous backing ones, we press them in, we pull them out immediately, and the needles are now embedded in the skin, as you can see, and the backing has no needles in it. So we accomplished this goal of having needles that don't require a shear. So you just press them in, the porosity causes the backing to collapse, and immediately you pull the patch away. So what we move to now is, having made some progress on these microneedles and achieved this 1 month delivery, we wanted to go longer still. We wanted to get out to a 6-month delivery. And the reason for that is not only that in surveys that have been done, studies that have been done to find out what people would like, women generally prefer 6 months over 1 month, more convenient, but also for a cost reason. We've been given the objective by the sponsors of this work, so USAID and Gates Foundation, that we need to be able to make these microneedle patches at a cost of $3 per year. That's what it costs in low-income settings. Women can get contraception for they may pay for $3 per year, or the government they pay for it, but anyway, the cost of goods is $3 a year. So we need to match that. And that's a pretty tough challenge. If we have to make 12 microneedle patches a year, that's more expensive than making 2 microneedle patches a year. Obviously, the 2 microneedle patches that have 6 months of drug, they each might be more expensive than making a 1 month, but not 6 times more expensive. So that's another motivation, is to try to meet the cost constraints and have a patch that's truly useful globally and not only for those who might be able to afford it. So that led us to wanting to deliver for 6 months, and to achieve that 6-month delivery, we're moving to a core shell microneedle patch. So for those who are in the drug delivery field, there can be a matrix design for a drug delivery system. There can be a reservoir design. So the matrix design is one where your drug and your polymer, they're all mixed together homogeneously, and you can get slow release from that. But if you have a reservoir design, you have the drug all in the interior, and then you have a film of polymer that surrounds the drug, and there's that rate-controlling membrane that film creates. That slows things down even more, and allows you to shift from first-order release, that is release that's initially fast and then tapers off and gets slower, to zero-order release, where the release is at a constant rate. Because you have this membrane that is a constant barrier, therefore your release of drug across that barrier can be constant, at least until the very end, when the drug concentration in your reservoir gets sufficiently low that then it tapers off. So, and another reason to use it is that you can load more drug. You can fill that drug in the core with lots of drug, and then have your thin membrane that goes around it. So advantages are we can get more drug per needle, we can achieve a longer release rate, and we can get that constant zero-order release. But there's some technical challenges to be able to do this in a microneedle. And so talk about that, how we're able to make the core shell structure. So the way we do it is we do a first cast of polylactic acid and do it in a centrifuge so that we can push down the solution that we have cast, and in that way we can get a film of the polylactic acid around the periphery of the mold, creating the shell structure of the microneedle. We then do a next cast, which has a mixture of PLGA with the drug levonorgestrel to make the core. So we switched from the PLA as the shell to PLG as the core because we needed to change solvents. If our second cast uses a solvent that dissolves polylactic acid, we lose our shell. So we have to now use a solvent that won't dissolve the polylactic acid, but will dissolve the PLGA and the levonorgestrel, and we can make the core with that. We then do a next cast, which puts a cap on the needle, so we want to completely surround that core with a shell, not just on part of it. So we completely surround it by putting a cast on. Then there's something we call funnel film. You can see there's kind of a funnel structure at the top that helps us with the manufacturing and the loading process. And we don't want to have that coated with the polylactic acid because then we're not going to get an easy separation of the microneedle. So we clean that off, and then we put on our backing, and we're done. So a little more complicated process here in order to get this core shell cap structure, but we think it's worth it, as you'll see. So now you can see an image of microneedles made in this kind of a way. You can see that the needle part looks cloudy. That's because there are all these levonorgestrel crystals that are embedded in there. And then the funnel region and below is clear because that is this is the water-soluble backing, and it doesn't have levonorgestrel crystals in it. And on this scanning electron micrograph, perhaps you can see the interface between the rougher surface with those crystals in it associated with the biodegradable polymer part, and then the smoother part that is the PVA sucrose backing. We did some confocal microscopy imaging. You can see a top view and a side view of one of these microneedles. The shell is a green color, and you can see it's more in the periphery. The green dye incorporated just into the shell cast. We added a red dye into the core cast, and you can see that there's this segregation of the core being in the middle and the shell being in the periphery. And finally, then, we did look at release from the system. So this is in vitro release, just in a saline environment. The black curve shows the cumulative release from a matrix needle. So that is, we made the core of the microneedle, but didn't make the shell around it. And you can see the first order kinetics and relatively rapid release on a month or more. The blue curve is now the core shell microneedle, where now we do have a steadier, more zero-order-like release, more constant release. And you can see it's extending out for in the neighborhood of 6 months. So it seems like the addition of the shell around the core makes a difference here and slows down the release process. And we have, in the meantime, now done a study in rats with needles like the ones that I showed. You can see the levonorgestrel level over a period of time. That's, at this point, is almost out to 6 months. Study is still going, so we'll see. And you can see that there's a small burst at the beginning, just in the first day or so. And then, yes, a little bit noisy. There are error bars here, but there's basically a pretty flat line of the concentration representing that zero-order release happening over the course now of approaching 6 months, about 5 months so far. And we have this target level for the drug that's shown with the dashed line. In knowing the pharmacokinetics in the rat and the human, this dashed line represents the level you want to have in the rat to correspond to the level you would need to have in a human. So a final topic that I want to mention is applying these microneedle patches to human subjects and designing the microneedle in a way that it will effectively go into the skin. And I'll say a little more about that. So we need to make our needles contain enough drug for a human. Rat is a whole lot easier because they're so small. And that means we need either longer, bigger microneedles and or more microneedles. Microneedles, by definition, are small. We want to load 6 months of drug, plus we want to load a bunch of biodegradable polymer to control the release. And there's just a limited amount of space to put all that material in. The target amount of drug that we need to deliver is about 10 milligrams, and we'll probably need about another 10 milligrams of polymer in order to have the human dose delivery occur. So we're concerned about if we start making more and bigger microneedles, will there be issues? Will there be pain associated with these larger microneedle arrays? Will the microneedles effectively penetrate? As you make more needles, you can get this bed of nails effect, and the needles just don't go in. What is our drug delivery efficiency? So are we getting all the needles to go all the way into the skin? And what is the preference? So what do the subjects think about the trade-offs of different microneedle designs? So we made 18 different microneedle patch designs. There's lots of detail here. I don't expect you to read it. But the point being, we varied various geometric features, numbers of needles, sizes of needles, length, width, and so forth. Made all these 18 different types of needles and then applied them to human subjects. And you can see here just examples from 4 of these microneedle arrays. The upper image shows what those needles look like. The lower images show what the needle looks like after insertion. Anywhere from on the left side, a lot of the needle dissolved, to the right side, where just the very tip dissolved, that meaning the microneedle didn't go in very deeply. You should note, these are fully water-soluble microneedles. They're not the biodegradable polymer ones. We want these fully water-soluble so we can easily see how deep the needle went into the skin by looking at how much dissolved. And the final is skin staining. This is where we have this gentian violet dye, and we can see how many punctures occurred. So the lower one is the puncture efficiency into the skin, and then the middle set of images is how deeply it went into the skin. Not just did it enter at all, but did it go deep. So then we made some measurements on these things. So the delivery efficiency, this is a measure of how deep the needle went. That is, what fraction of the needle disappeared. What fraction of it dissolved in the skin versus what fraction of it was still there when we pulled it out afterwards. And you can see we get a delivery efficiency as high as about 80%, and as low as even less than 10% for the one that was particularly bad. So it really does matter how you design your microneedles. We also look then at the total dose delivered. So these are not the same thing. Delivery efficiency is what fraction of the individual needle delivered. Total dose delivered accounts for the fact of how many needles do you have. So if you have a lot of needles that are reasonably efficient, that could be as good or even better than if you have very efficient needles, but you didn't have very many of those needles. So there are these trade-offs that exist. And we look at the ones that are the winners. You can see design 3 and design 18 were the ones that were able to deliver the largest dose. Because at the end of the day, that's what we really care about. Efficiency is nice, but the dose delivered is what we need. And so the dose delivered for these designs, you can see, is 4 to 5 milligrams. So it's in the neighborhood of where we want to be. We then asked subjects what they thought about the experience of having these patches pressed into them. And the pink bar is the percent who said it was painless. The blue bar is the percent who said it would be acceptable. So that is, it would be acceptable to them to have this kind of a patch applied every month or every few months. And you can see that the pink bar, it varied in terms of people generally. There were some people who felt some level of pain, but a very low level of pain, but nonetheless they could feel it. But you can see that in most cases it didn't bother them. In most cases they said it was acceptable. And so here's this microneedle design 3 and 18. Those were the two that were best for dose delivered, and so 3 is what we like best here. Everyone thought that was fine with them, and the delivery was highest. We also looked at the skin response, so the erythema of the skin. Everybody had some erythema right after the administration. The lower sort of grayish bar, that represents the percent who have the grade 1 erythema, so the mildest, and you see a picture of what represented a picture of that. Some people had the grade 2, which is a little more erythema, mostly grade 1, in all cases transient. And again, no safety concern from something like this. So finally then we asked the subjects, what do you think about this whole thing? So we asked them, if you had a choice, and I should note these are all women of contraceptive age, I think all Georgia Tech students, so it's a limited population, does not represent what the whole world might think, but at least what this population thinks. 10% said they prefer to have the daily pill, but 90% said they would rather have a microneedle patch like they had just experienced as opposed to a daily pill. We can then compare it to, would you rather get an injection every 3 months or use the patch? Everyone wanted the patch. We said, what about a 3-year implant? Well, some people wanted that, but still 80% thought the patch would be better for them. What about a 3 to 10-year IUD? There, a few more wanted that, but the majority still preferred the patch. And I should say our goal here is not to convince everybody to have the patch. The question is there some reasonable subset of women who might like this? And so this suggests that there is. We asked about the duration, and there was a strong preference for longer patches, 6 months being most preferred. And then we asked how they would want it administered. Half said they'd like to self-administer it. Half said they'd like to self-administer it the second time. But the first time they would want some help from a healthcare provider. There was a third option if I want the healthcare provider to do it every time. Nobody chose that option. So that brings me to the end of the story. And I've tried to describe to you this microneedle patch and what this technology might be capable of. We spent a fair amount of time talking about a really critical issue for us, which is needle separation and different strategies, whether it's a bubble or an effervescence or porosity, in order to facilitate that separation and make it convenient and reliable for someone to self-administer. Of course, the drug release was a focus as well, and where we're working mostly these days on the core shell geometry that I've described to enable this long-term release out to 6 months. And we have looked at this issue of usability and acceptability by human subjects, because at the end of the day, we're developing drug delivery systems for patients to use, so we need to know what patients think so that we can design not only for technical objectives, but for ultimate success in a clinical environment where real people are using them. So let me thank the research group that's done the hard work to generate the data that I have the pleasure of sharing, and I'd be happy to take questions with whatever time remains. Thanks.
Thank you, Mark, for this very insightful talk and excellent technology. So the floor is open to questions. Parveen.
Yeah, very nice talk. Very nice question, actually. So after you put the microneedle, what happens inside the skin? Is there any reaction or there be any clotting afterwards?
So the microneedle gets embedded in the skin, and then we think the key thing that happens is it contacts with water in the body. And so that either leads to a dissolution, or in the case of a biodegradable polymer, leads to the biodegradation process. So that's the functional side, is basically the microneedle contacts water. From the more biological side, you've caused a micro-injury to the skin. You've punctured the skin. And so there is a response to that, and so that's the erythema response that we see. There's, and what I've been showing, all the human results, those are all with the water-soluble microneedles. Our goal is to have these biodegradable microneedles in the skin that don't dissolve and disappear in hours, but stay there for 6 months or more. So we are gearing up to do a formal human study where we see what happens when the needles are embedded in there and how the body responds. We have some preliminary data, including in myself, as well as a few other people, where we've applied these microneedle patches. The anecdotal observation so far is that for the first week, you see just very faint redness or erythema, like what I showed, very faint. But after a week, you can't even tell where the patch was applied. I'm out to a little over 2 months at this point, and I mean, I know where the patch was applied, because I know where it was done, but you can't tell at all. So it looks like the skin's response after just the minor injury of the puncture is a very good one. But that's going to certainly depend on the material that you use. And we don't have, I didn't give myself contract performance. So it's possible there could be a response to the levonorgestrel being released. But levonorgestrel is released in subcutaneous implants for years, and that's tolerated very well. So we're optimistic about that.
And another small question. So this 6-month long process, you also said that it's a very noisy data. Why was it like that? Why there was so much fluctuation?
I think it's a combination of things. One of them is that as soon as you go in vivo, different animals, different people, they're different. And so in vivo data is always going to be noisier just because of that inherent variability in biology. But another reason is the noise, if you look at the individual rats and follow their pharmacokinetics, for an individual rat it's not as noisy except at certain points. So you'll see a relatively well-behaved curve, and then there'll be a little jump to a high level, and then it'll be behaved, and there'll be a little jump to a high level again. So we were curious, is this an artifact, or is this real? Is this something about the sampling or something about analytical methods? And we, I guess unfortunately, suspect that it might be real. And I think that it might have to do with this core shell structure. That as long as the shell is intact, we have a good release. But if the shell is not intact, there can be a more rapid release that occurs. And so we have some concern that it may be that at various time points, some needles are failing. And so it's suddenly dumping the drug out, and you get these little jumps associated with that. So the jury's still out, I don't know, but that's a possible interpretation. Fortunately, the therapeutic window, so that is the difference in concentration between effective and toxic, is very large for levonorgestrel. So we should be able to accommodate some of this variability, just we don't want to go below the therapeutic level, of course.
So just in continuation, have you done any studies to compare the oral contraceptive pharmacokinetics, the 3-month injection, and so on and so forth with the microneedle patch?
We will be doing this fall a study in mini pigs, and in the mini pig study we will be comparing these microneedles to a conventional injectable slow release formulation. So we'll have that direct comparison, and our goal would be to have very similar pharmacokinetics to that, just a steady release over months. In terms of the pill though, we're definitely different. When you take a pill, the concentration goes high and then low, and then the next day the pill goes high and then low. So we should have definitely a steadier concentration than the ups and downs inherent to pills.
Very nice talk and really useful application. So just as a general query, whenever we have something which is inserted into skin, we have back pressure of blood or something. Apart from that, those who are doing exercise or rigorous things, sweating will be there as one of the issue. So how stable this patch will be for long run? Apart from that, we also like normal flora on the body. So is there any possibility of infection when you press it?
I think you're emphasizing things that are on the surface of the skin. So that is definitely important at the time of application. The patch wear time is short. It's anywhere from a second to minutes. And so the time that what's happening on the surface matters is short. So you press the patch on, you take it away. I'll come back to that in a minute. Once the needles are embedded, if there's any sweating or if something comes on your skin, it should be irrelevant because the needles are embedded in the skin is healed up, close it off, and it's really an implant at that point. It's not a patch. At the time of application, though, these things do matter. If there's sweat on the skin or dirt on the skin, I mean, you do want to clean the skin off first, and that would be a standard kind of a procedure. There is the potential issue, which maybe you're thinking of, that there could be an infection that results and you're breaching the barrier, and there could be a problem with that. As you may know, the real danger of infection associated with conventional hypodermic needles is if the needle is dirty, say it was reused. The source of infection or contamination is not something on the surface of your skin already or the residual hole and something going in. I mean, we have nicks and cuts in the skin all the time, shaving, playing football, whatever. You get these minor injuries and don't get infected usually. So as long as, the lesson from that, as long as the microneedles themselves are sterile and the skin is basically clean, we don't expect to have an infection problem. The anecdotal data from work that we've done and others have done as well has never shown any kind of an infection coming, so time will tell, but the expectation is that that shouldn't be a significant risk.
And just last curiosity, like which factor decides the dissolution of that particular material? And is it common for all the human beings? Or is it vary based on pH or whatever other conditions?
So the dissolution, if it is a true dissolution process and not a degradation process, then the dissolution is really water. And we all have water in our skin. Hydration levels can vary to some degree. The dermis could be 60%, 70%, even 80% water, but it's still mostly water. So the dissolution should be quite similar person to person. If it's a degradation process, well, if it's hydrolysis, again, it's water. But some polymers may be degraded by an enzyme, and so there may be greater variability in enzymatic levels in different people. But that's going to become very specific to the formulation you're using.
Mark, thank you for that fascinating talk. I had a question which had to do with also what he addressed, but I suppose you have covered it, which is got to do with the 6 month, do any of the needles fall off or do they get rubbed off? Especially if you're looking at low income countries, you would expect to see a lot more in the hotter areas, sweat and so on. I think you've addressed it. But I have another question which has got to do with actually the methodology that you followed, and this will be good learning for some of our students as well. you had a very nice segment on the problem definition itself, the need identification, defining what is needed, and scoping it out in different ways, including the 6 months and so on and so forth. And of course, maybe your funding agency also contributed to setting those goals. And then, of course, you went on to design it and then implement it with the tools which are available and demonstrate the result. I wanted you to talk a little bit about what are the lessons in need identification and problem definitions, what in such kind of problems, because that is very often a large part of the exercise, right? And how do you do that? And are there some practical pointers, tips for students who are in the process of developing such products?
Yeah, I think this is a very important point. I think any scientific or technological challenge, solving it is hard. We all know. Doing research, it's hard. It takes a long time. It takes a lot of effort. So I think it's really important to spend a lot of time up front to decide which problem you're going to solve. Because whatever problem you solve, it's going to be a lot of work. So you want to make sure you're solving the best problem and using the best approach you can to create a real solution to that problem. So yeah, I did spend time to do that problem definition, and some of that does come from, fortunately, we have funding agencies that are very much aware of these things. We're in Atlanta, the Centers for Disease Control and Prevention is located there, so we get a lot of good advice from them. The Emory University has a medical school, and we collaborate heavily with the medical school. So I think it's really quite important to talk to people who have the additional expertise needed to understand the context for the technical challenge that you're working on. Because I know for myself, and probably most in this room, we have a limited expertise in a certain area, and probably more to the basic side of things. But if we're going to use those basic skills to solve problems that ultimately really solve problems, not just make papers, and there's value in good scientific papers, but if your goal is to have that scientific paper be leveraged to solve an industrial societal problem, then talking to the people who know about those things, I think, is really very important, and to do that early. Because once you set your objectives and get to work on it, it's much harder to go back later and change those objectives. So doing that homework up front, I think, is worth it.
We'll have 2 more questions.
Hello. Thank you for your excellent talk. So just question about your controlled drug release delivery. So you say the delivery, the drug get released up to 6 months. Or 3 months, or whatever you can control it. Does the efficacy also remain for 6 months, or does it start to taper off before the end of 6 months? And is there an exact cutoff at 6 months, or it can continue even beyond 6 months? And second small question following up on that, do you need the skin to be in a specific condition before applying? That is, you have to clean it, or anybody can use it just by brushing off to be okay? Thank you.
The ideal release profile would be that you keep the drug concentration exactly at some target level, and then at the end of 6 months, or whatever your time is, drops off and you're done. That's, of course, very hard to do. But there is a minimum level that you need, and so you definitely want to stay above that. If it dips below that at any point, then you're in danger. Then you're not at an effective level. The woman could become pregnant. So you definitely want to stay above it. And then the other constraint is not too far above it, because then you can have toxic effects. So as I mentioned, that window between minimum effective concentration and toxic is pretty wide. So we'd like to be well above that minimum, just to make sure with the variability we never dip below. There will be a taper-off region, by definition. And actually, if we have a 6-month patch, we actually want to deliver for 7 months. Because people are unreliable. And at 6 months, maybe you don't get around to getting the next patch till 6.5 months. So we do want to have a buffer to go a little bit longer, but then we do want it to taper off quickly, because women taking contraceptives oftentimes would like to stop and have a child at some point. And so we don't want to have people wait a long time. So the taper off period is important. We're only at 5 months in the data that I showed, so we'll see how long that taper off is. But my guess is there'll be at least a couple of months of that taper off period where you're kind of in limbo. You're partially protected, but not fully protected. So yeah, that I think is going to be inherent. In terms of skin preparation, the skin should be clean. Probably an alcohol wipe first will be a good idea, although in most of the world, WHO recommendations, for example, is before giving a hypodermic needle injection, an alcohol wipe is not suggested because there's no evidence that it actually helps. In the US, where we have too many lawyers, everyone gives an alcohol wipe just to be safe. So I don't know, maybe we'll give an alcohol wipe, maybe not, but I mean, you definitely don't want to be dirty.
Last question, unless there's any student who wants to ask a question.
Hi, nice talk. So my question is, you haven't talked about the drug property, how that affects the microneedle. Like you just talked about one drug, but if you have a drug with, highly hydrophobic, if you want to really generalize the technology, how is the drug plays a role when you make that microneedle?
Yeah, no, the drug definitely plays a role in the manufacturing process that you choose, as well as the ultimate properties of the microneedle. Just briefly mentioned for the ultimate properties, having all these levonorgestrel crystals in the biodegradable polymer matrix weakens the microneedle. You've got lots of interfaces. And so as a result of that, the needle is weaker. So that is a challenge for us. We have to make sure the needle is strong enough, even with all of those crystals in place. For the manufacturing, as a solvent cast process, you have to have a solvent, and that solvent has to dissolve whatever it needs to dissolve. So if we have levonorgestrel and polylactic acid, those are both hydrophobic materials, and so there's a variety of organic solvents that you could use in order to do that. But if you wanted to embed a hydrophilic drug in your hydrophobic matrix, well, now you may not have a common solvent for them both. So then maybe you have to do a suspension where your drug is suspended in a solvent that dissolves the polymer, or maybe you'll make an emulsion, so your drug's in the aqueous phase and a continuous solvent. So you'll have to do some design in order to accommodate the solubility issues.
If no students want to ask any question, then let's thank Mark for this thought-provoking talk. And before we close, I'd like to request Dr. Prerna to present Mark with the memento.
First, the book. Yeah. This is for your information. Done? Yeah. This is a book about NCL.
Yeah.
It is written by the illustrious chemical engineers from here who went on to become a professor. Was it Iowa State? Yeah. Okay. And he was the head of the chemical engineering department, went on to become the director of NCL. And it's a story about NCL's history, going up to maybe about 10 years back. And this is a painting by one of our scientists who is watercolor done about NCL. So, and as I was telling you, the first director lived here. Well, thank you very much. Very fascinating talk. Thanks for taking the time out. Professor Prosnis is spending some time in ICT Mumbai for a month, and he is doing some work with the Serum Institute, so we were happy to have him here, and I hope you'll come again. We'll have a chance to have a discussion again. Thank you. Thanks a lot.
And I'd just like to thank the event management team for making this event possible. Thanks.
Thank you for joining us. The scientists, if anybody is interested in, we are having a greet and meet session at the boardroom, right? So some of you can join. I've already sent to some of the people who work in this field, but anybody else wants to as well.